In this set of lessons, we'll look at the biological basis of learning and memory. The consensus view in the neurosciences is that synaptic plasticity is the key mechanism for learning and memory. Now, in previous lessons, we learned about system consolidation, the idea that memories are reorganized over time. And whether it's the standard consolidation theory or trace transformation theory, both theories require that some kind of changes are happening within neurons in the hippocampus and the cortex so as to store a memory trace of an episode or of some kind of information. The idea that there are biological changes that underlie learning and memory goes back a long time, all the way back to the pioneering work of Ramon and Cajal, who hypothesized that the bumps on the dendrites of a neuron were connection points with other neurons, where changes associated with learning might occur. And of course, today there is much evidence that Cajal was correct. Learning and memory are rooted in changes at the synapse. Now, if we take a look over here, some of his fantastic drawings here, we see two neurons here, the cell body, axon. We got an extensive dendritic tree, and you can kind of see the little bumps on the dendrites here. Those would be spines. What we don't see are the axons and terminals making synapses on those spines. But Cajal was an expert microscopist and neuroanatomist, and he speculated that learning involves biological changes at certain sites on the dendrites of neurons. Decades later, in the mid-1900s, scientists were gathering more evidence that learning involves physical changes in the brain. Take one group of rats and raise them in an impoverished environment, another group of rats, raise them in an enriched environment, and then look at the brains of those rats later on, you find significant differences. In rats in the enriched environment, you have a thicker cortex, larger cells, more extensive dendrites, more dendritic spines. It's as if the extra learning that was happening in the enriched environment was causing physical changes to neurons and their interconnections. What extra learning is happening here? Well, you have a lot more social interaction here. There are things to learn about other rats, lots of toys to explore. In the impoverished environment, no social contact and no other toys to learn about. So the suggestion was that learning does indeed involve physical changes in brains. Also in the mid-1900s, a Canadian psychologist by the name of Donald Hebb articulated what is today still sort of the basic understanding of what's happening during a learning process. He said that when axon of cell A is near enough to excite cell B, or repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency as one of the cells firing B is increased. Today, it's often summarized as cells that fire together, wire together. So at this point then, we can start to put some pieces together. We know from the case studies of HM and others, the hippocampus seemed to be a critical structure in forming memories. And at the same time, it was understood that perhaps physical changes were happening between neurons in the brain, and this accounted for memory. We'll call those physical changes at the synapse synaptic plasticity, referring to the capacity of synapses to change their strength. So perhaps what was going on is that when we undergo some kind of learning episode, physical changes, synaptic plasticity is happening between neurons in the hippocampus and other regions, and long-lasting changes then would be the basis of a memory. We can apply the word consolidation here as well. Synaptic consolidation then would be the fundamental process that converts a learning-induced change in synapse strength into a long-term memory. In other words, if you increase the synapse strength and that increase in strength persists for a long time, that then would be the biological basis of a long-term memory. Notice consolidation we learned before in the system context, where system consolidation referred to the reorganization of a memory across different brain systems, but still in both of these systems, the changes that would be happening at the level of neurons would be synaptic plasticity. So we would have synaptic plasticity among hippocampal neurons and synaptic plasticity happening among cortical neurons. So in the remainder of the lesson then, we'll take a look at the hippocampus in more detail. Recall the hippocampus resides underneath the temporal lobe bilaterally. Here's a cross-section of the whole brain showing the location of the hippocampus, and here a section of brain has been stained to see cell bodies. Take a look at higher magnification. We can see some structure of the hippocampus. We have fields of cells, the dentate gyrus and CA3 and CA1 cells. In higher power magnification, we can start to see individual cell bodies. Now, a breakthrough occurred in the latter part of the 20th century, in the late 1900s, where scientists were able to make thin slices of the hippocampus and keep the tissue alive in solution. So the slice would be bathed in a nutrient solution, and you could keep this slice alive for up to 6 hours or so, whereby you could then play around with stimulating and recording electrodes. So you could gain control over the nature of neuron communication within the hippocampus. You could have stimulating electrodes that would stimulate axons to generate action potentials, and you could have recording electrodes either in the population of target cells or recording the electrical activity of one target cell only. So this experimental arrangement led to some fundamental breakthroughs in our understanding of synaptic plasticity. A very important discovery was made in the 70s with hippocampal cells. Let's imagine here are two hippocampal cells, the presynaptic cell and the postsynaptic cell. Now, imagine that in this pathway for the presynaptic cell, we just give a small test pulse. So we activate the cell so that it makes one action potential. Well, it arrives at the terminal, some neurotransmitters are released, and they bind to receptors, and ions will enter, and you'll get some kind of small electrical response, a depolarization in the target cell. Now what they did was, to that same neural pathway there, they stimulated the presynaptic cell at high frequency. So lots of action potentials racing down. We're going to get lots of neurotransmitters released, a lot of channels opened up, and a lot of ions, sodium ions, rushing in, and we will pass threshold, and the target cell will now generate its own action potentials in response. But here was the fundamental discovery. If then, a moment later, you go back and just deliver one test pulse, one action potential down that same neural pathway, what they found was an increased response in the target cell compared to the original cell. Right? So it's as if this high frequency stimulation of this pathway that generated activity in the target cell, it's as if that activation somehow increased the strength of the synapse. And this strengthening of the synapse became known as long-term potentiation, because the enhanced strength of the synapse lasted for many hours. And it was potentiated. The synapse was potentiated, it was increased. Now, what were the possible mechanisms of an increase in the synapse strength? Well, if we just think about a basic synapse, we have the axon and the terminal, we have neurotransmitters, we have receptor proteins on the spine membrane here, and those are channels that allow ions like sodium in. So this is the nature of synaptic communication. Well, how could a synapse get stronger? Well, there were a couple possibilities. One possibility is that, here's the synapse before LTP, long-term potentiation, is induced. You got a certain amount of neurotransmitter released per action potential, and you have a certain number of receptors. Well, perhaps after long-term potentiation was established, for each action potential, maybe more neurotransmitters were released. And if there's more neurotransmitters released, you'll have a larger kind of signal in the target cell. Or maybe more receptors were on the target cell membrane. So the spine might have more receptors. With more receptors, you'll get more current going into the spine, so a greater synaptic efficiency. So those would be two possible mechanisms for increasing the strength of a synapse. There's a third as well, and that is structural changes at the synapse. That scientists noticed that some spines were smaller, some spines were thicker, some were more narrow, some had larger surface area. So the structure of the spine itself, the morphological shape of the spine, might be one way to modify the strength of a synapse. So these are three possibilities early on: increased neurotransmitter release, increased number of receptors, and physical structural changes at the synapse. After establishing the basic phenomenon of LTP, long-term potentiation, scientists went on to characterize the conditions under which LTP occurred at a given synapse. There are three conditions here that we'll take a look at, and then we'll draw some conclusions. Under the first scenario, what scientists did was they stimulated with high-frequency action potentials the presynaptic cell, but while they were doing that, they prevented the target cell from generating action potential. So they hyperpolarized it. They injected chloride ions, negative current, into the cell to prevent it from responding. Under these conditions, you do not get the LTP phenomenon. You do not strengthen the synapse. It's as if the target cell must fire in order for a synapse to be strengthened. In scenario 2, we stimulate the presynaptic cell again with high-frequency action potentials, but it's a weak pathway. It would be insufficient to drive the target cell to make its own action potential, so the cell won't reach threshold with this stimulation, but we come along and we inject extra current, so we can reach threshold now. When a weak pathway is stimulated and we stimulate the target cell, yes, indeed, you do get LTP. You can strengthen this weak synapse. Condition 3, if we simply stimulate the target cell by itself without stimulating the presynaptic terminal, you do not get LTP. In other words, this suggests that you need the presynaptic terminal to be actively taking part in firing the target cell for LTP to happen. In other words, if you take 1, 2, and 3 together, this is support for Donald Hebb's suggestion that neurons that fire together wire together.